Welcome, everybody, to this special News of the Day edition of Star Cells and God. This is the podcast where we explore how the latest discoveries in science provide evidence for God's existence, the reliability of Scripture, and the credibility of the Christian worldview. My name is Rana. I'm a biochemist and a Christian apologist, and I work for an organization called Reasons to Believe. This is the organization that sponsors this podcast, thanks to the generous gifts and donations of viewers and listeners like you. If you want to know more about how you can support the work of Reasons to Believe and Star Cells and God, go to our website, reasons.org. Also, at the website, you can learn more about our organization. Also, please follow us on social media, RTB_official. And then last but not least, make sure you go to our YouTube channel, Reasons to Believe, and subscribe. There you can gain access to all kinds of great video content exploring a wide range of questions relating to science and the Christian faith. Also, while you're there, use the notification button so that you can be alerted the next time a new episode of Star Cells and God drops. Okay, with all that under our belt and without any further delay, let's go ahead and take a look at this headline-grabbing discovery that we want to discuss on today's episode of News of the Day. Late last week, there were news items that were all over the world announcing a breakthrough by a team of researchers from Tianjin University in China. And these researchers claimed to have been able to control robotic systems using human brain tissue that they cultivated in the lab. And of course, this is a really bizarre headline, right, that evokes all kinds of provocative questions, some relating to the ethics of the work, some relating to the safety of the work. The bizarre nature of it also, again, evokes a lot of questions as to what precisely did these scientists do. Questions that we might summarize with this singular question, should we play God? And how, from a Christian worldview perspective, should we view this kind of technological breakthrough? Now, I wasn't able to find the journal article that this headline-grabbing discovery created or generated. I'm sure there is a journal article that has been published. I just simply have not been able to track it down. However, all I've been able to find is a press release from the university in China, the Tianjin University, but I believe that the work that's reported on is credible work, simply because, number one, Tianjin University is a highly respected institution. Number two, this research team has published other work where they have looked at lab-grown human brain tissue, characterizing it and exploring its utility for a number of different applications. So there's no reason to think that this work, which is probably an extension of this larger research program that they're involved in, is not credible or legitimate. But in absence of having the actual bona fide journal article in front of me to be able to break down, what I thought I would do is give you as viewers and listeners a little bit of an overview conceptually about what these researchers were trying to do before we start unpacking some more profound philosophical and theological questions centering around this work. And this is a slide that I was able to find in another journal article by researchers that were doing similar kind of work. In fact, the work done by these Chinese scientists, though bizarre as it may seem, is actually work that's being pursued by scientific groups all over the world. And this is a part of a larger umbrella of a research project called biocomputing, where the goal is to create the next generation of computers and electronic devices that are actually controlled through the integration of biological systems into computer hardware. And specifically, the work that this research team is engaged in is called organoid intelligence, which is an analog to artificial intelligence. And what they're doing is that they're growing these three-dimensional cell cultures in the lab that in effect are mini brains. They're technically called brain organoids or cerebral organoids, which are again these three-dimensional cell cultures that are somewhere between about 0.5 centimeter to 2 to 3 centimeters in diameter, and they are integrating them into a computer chip, and then by connecting electrodes to the brain, they're actually able to extract electrical signals from the brain organoid and also introduce electrical activity into the brain organoid, send electrical signals into the brain organoid, and the network of neurons that are part of these brain organoids are actually used to process that information through machine learning algorithms in such a way that these brain organoids can essentially produce appropriate outputs given particular inputs, and as such can then be utilized to not only interface with electronic devices, but actually be used to control robotic systems. So that's what these researchers are doing in a nutshell. And again, this raises all kinds of questions about what specifically are these researchers doing and why would they engage in this type of work, and is this work ethical? Is this work potentially dangerous? Again, should we play God? And before I get to that question, what I'd like to do is spend a little bit of time talking more about the science and the technology that undergirds the work that's taking place in organoid intelligence. And it's been my experience that when people encounter bizarre technological breakthroughs like the one that's been reported just recently by this team from China, oftentimes they react with concern or with fear that is actually unfounded. And that concern and fear reflects a perception about the technology that doesn't necessarily match the reality. And so what I thought I could do is help to explain, again, the science and the technology that undergirds this work, talk a little bit about the strengths and the limitations of this work. Why are people pursuing this work? What are the challenges that remain? And from that standpoint of understanding, I think we're going to have a better capability of appropriately assessing the ethics and the safety of this work as we address the question, should we play God? But also, I think we're going to have a better capacity to appropriately engage the critical issues that this technology centers on in an intelligent way, in a relevant way that actually helps us to advance the Christian worldview and our contribution to the larger project of helping to guide technological development in the years to come, not only in terms of what technology we should pursue, but also how we should utilize different technologies. And so in terms of understanding the science of organoid intelligence, the place to start is actually with the anatomy of a neuron, right? Because in effect, what the researchers are doing is taking advantage of the neural networks that naturally will form in these brain organoids and using these neural networks as a way to store memory or store information in the form of memory, and then also to engage in processing through the neural networks that exist, again, inside the brain organoids. Very similar to the neural networks that computer scientists will utilize for artificial intelligence. And so what I have here is a cartoon showing the anatomy of a neuron, because the neurons are the cells in these brain organoids that are ultimately the centerpiece of the technology. And a neuron consists of a cell body, which is shown here as roughly an octagonal-shaped structure that has a nucleus in the center. And extending from the cell body are these tree branch-like structures that are called dendrites. Also extending from the cell body is a very long, thin projection called an axon, and that axon is wrapped intermittently by what are called myelin sheaths, which are layers of fat. And in between those myelin sheaths are these little nodes called the nodes of Ranvier. And then the axon terminates in a tree branch-like structure that each branch point culminates with a bulb called a synaptic bulb. And so neurons, again, are the cells in our brain that form interactions with other neurons to create these neural networks. And these neural networks are what is used to interpret the information that our senses send to our brain. When we hear something, when we taste something, smell something, see something, that information is sent to our brain processes that information, and then acts upon that information, whether subconsciously or consciously through motor operations that control the different parts of our body. Also, we process that information, and some of that we store in memory within our brains, others that we begin to reason about through our cognition, our cognitive capacities. So all of this is essentially information that our brain takes in and processes and then determines what to do with because of the neural networks that exist inside the brain, where the neurons are interacting with other neurons. And these neurons, as they interact with other neurons, can receive signals from adjacent neurons. And so these signals are in the form of chemicals released by adjacent neurons in areas called the synapse. And these chemicals will bind receptors that are proteins embedded in the membrane of dendrites that will then trigger an electrical signal that will travel from the cell body down the axon to the synaptic bulbs, and there chemicals will be released that will then impinge upon adjacent neurons. And in effect, what you see with neurons is they're responding to chemical signals from other neurons, and in turn initiating an electrical signal along their axon that transmits the information, again, from dendrites to the synaptic bulbs, and communicating in turn with other neurons that this particular neuron in question is interacting with. And this electrical signal that is passed down the axon, along the axon, is due to what's called a polarization of the membrane region of the axon. Cells are surrounded by a cell membrane, which consists of a bilayer of a structure that looks like a sandwich that consists of two molecular layers. And it turns out that there are proteins that are embedded in the axonal membrane that can pump potassium ions into the interior of the cell and pump sodium ions to the exterior. This is a sodium-potassium transporter or pump. And as a result of that, there is a higher level of potassium on the inside of the cell than on the exterior, in the exterior environment, and a higher level of sodium in the exterior environment compared to the interior environment. And that mismatch in concentration of sodium and potassium sets up an electrical charge across the membrane, where I believe the interior of the membrane is negative relative to the exterior. And so when a chemical signal impinges on dendrites, it causes a depolarization of the cell membrane, where now these sodium and potassium channels in the membrane of the axon open up, allowing potassium on the inside to flow to the exterior environment, causing sodium in the exterior to flow into the interior. This is a depolarization of the membrane, and in fact, there will be a point where there becomes a hyperpolarization of the membrane, where the interior now becomes positive and the exterior becomes negative, and then the sodium-potassium pump will kick in, restoring the resting potential across the axonal membrane. But what ends up happening is that this depolarization doesn't happen across the entire membrane all at once, but happens in regions, and it flows from one region of the axonal membrane to the very end, close to the synaptic bulbs. So this is a diagram showing the depolarization and the traveling of that depolarization along the axonal membrane. And so in a sense, what you can think of is that the neuron is essentially a cell that is capable of responding to chemical signals, initiating chemical signals, but then is able to conduct an electrical current. And so that when one neuron interacts with another neuron through what's called a synapse, which is where the axonal bulb interacts with the tips of the dendrites, that this is in effect forming an electrical wire that would exist within the brain, right? And in fact, what you see with neurons is that they're somewhere between 86 to 100 million neurons in the human brain that are networked into these neural networks where particular neurons are interacting with other neurons. And it's the patterning of the electrical activity in those neural networks that essentially is the way in which information is stored and information is processed by the brain, right? And so what the researchers want to do who are creating these systems that are, again, controlled by these brain organoids is to take advantage of this enormous processing power of the human brain on a very small scale, integrating these little brain organoids into chips that then allow them to control things like robotic systems. And the way in which they do this is that they grow in the laboratory these brain organoids. And this is a slide showing how these brain organoids are actually grown. They're grown from what are called pluripotent stem cells. And these are cells that can develop into any of the 210 different cell types in the human body. And depending upon the cultural, the media conditions of the stem cell culture, depending upon the types of growth factors that are added, you can convert these pluripotent stem cells in the laboratory into, again, the different cell types of the human body. Now, there are two sources of pluripotent stem cells. One are from human embryos. These are called embryonic stem cells, and these are generated when an egg cell and a sperm cell are used to form an embryo through in vitro fertilization. After a few rounds of cell division, those resulting cells of the early embryo are called blastomeres, and they are pluripotent stem cells that can be isolated by destroying the embryo and then cultured on a petri dish, awaiting conversion into different cell types. Now, this source of stem cells is ethically problematic for many people like me who hold to a pro-life position, who see an human embryo as equivalent to a human person, that here we are creating human beings to destroy them for their cell parts. And so this is, again, morally impermissible. It's morally repugnant. But there's another source of stem cells that more and more researchers are utilizing that don't have that ethical and moral baggage associated with them. These are called induced pluripotent stem cells that are stem cells taken from fibroblasts originally, which are skin cells found in adults, and are converted through certain types of exposure to growth factors and other transcription factors into stem cell-like states that are called an induced pluripotent stem cell, and that then can be converted in the lab into a wide range of different cell types. This is a non-problematic source of pluripotent stem cells because they're not coming from embryos, but from adult tissues. Once in these pluripotent stem cells have been cultured in a petri dish, a small sample is taken and they are grown in a liquid medium to form like a ball-like structure that's called an embryoid. And that ball-like structure is then transferred into a gel matrix where it begins to grow and develop into a three-dimensional cell culture. What's also critical is at some point that three-dimensional cell culture is transferred from the petri dish into a spinning bioreactor that helps to form the three-dimensional structure. And the net result is this brain organoid that is, again, somewhere between about 0.5 and about 2 to 3 centimeters in size. It will consist of about 100,000 neurons, as well as a number of other cells that are found in the brain. like astrocytes and microglial cells that play a support role for the functioning of the neurons. But in these brain organoids, these neurons will naturally form into neural networks. People are creating these brain organoids as a way to study the earliest stages of brain development in human embryos. These are helping us to develop a basic science understanding of, again, not only brain development, but how the neurons are functioning in the brain. And so these are very important research tools that have been recently developed, and people are exploring all kinds of biomedical as well as biotechnology applications for these brain organoids. Now, one of the limitations of brain organoids are the size, that they can't really grow much beyond about, again, a half a centimeter to 2 to 3 centimeters in diameter, because there is no way to get nutrients to the interior of the brain organoid. The nutrients have to essentially diffuse into that three-dimensional cell culture, which means cells on the surface are going to have ready access to nutrients. They're going to be readily able to get rid of cellular waste simply through diffusion. But as you move into the interior of the cell, it becomes harder and harder for those cells to gain nutrients and harder for them to eliminate wastes, because there's no vascularization. There's no blood vessels feeding the interior of these brain organoids. So this is a limitation right now with respect to these organoids. They can't get much larger than that. In fact, they can live for about 2 to 3 months before they eventually die, and they often will die from the inside out. So the idea here, again, is that these brain organoids, because of the neural networks, are going to function to some analogous degree like brain tissue, and can then be utilized in terms of their processing power and their capacity to store information. And it turns out that there is a technology that computer scientists use called neural networks. And these neural networks are basically modeled after the neural networks in the brain. And the idea here is, again, the neuron is like a microprocessor, where it is able to take signals from adjacent neurons and send signals to adjacent neurons and conduct electrical current. That is, you can think of them as a microprocessor. And so people have built microprocessors modeled after neurons, and then have integrated them into these networks of microprocessors. And this is called a neural network. That is an important tool today in artificial intelligence, where there can be information that's input into these neural networks, and those electrical signals will travel along different paths within the neural networks. They're going to generate an output, and through machine learning algorithms, you can actually train the neural networks to take particular inputs and produce particular types of outputs, right? And so the idea here is, again, instead of using these electronic neural networks, researchers are actually using these brain organoids as, again, a biological version of neural networks to supplant the electronic versions of neural networks that they're working with. And by introducing microelectrodes to the surface of the brain organoid, electrical information can be sent into the brain organoid and be processed by the neural networks and generate an output that can be collected by those microelectrodes. And again, using machine learning algorithms, the neural networks in these brain organoids can be trained to control things like robotic devices based on the input that the neural networks receive. Now, the reason why scientists want to do this is because the brain has incredible computational capacity because of the neural networks in the brain, has an incredible capacity to store information, but does so in a highly energy efficient way. The human brain can operate with less energy than it would take to light a light bulb, whereas the neural networks that are being produced for AI applications are becoming larger and larger and larger, and are beginning to consume greater and greater amounts of energy, to the point where we're about at the capacity of neural networks to function given the intense energy demands that they require for their operation, which caps our capacity to develop AI systems. So researchers are looking at utilizing brain organoids as a way to overcome this energy limitation. So that's one reason why organoid intelligence is being explored as an emerging biotechnology under the umbrella of biocomputing. Now, as I mentioned, people are also using brain organoids as a way to understand some basic information about how brains develop and how the neurons in the brain function, and the other cell types in the brain as well. And so by creating these organoid intelligence systems, that becomes another means by which we can probe the operation of the brain more directly and more deeply. And then finally, there are people who think that these brain organoids could one day be used to treat people with neurodegenerative diseases or with brain injuries, where these brain organoids could be integrated into the human brain to help overcome the loss of brain tissue because of injury or disease. And by creating these, brain organoids on a chip, it's possible, again, to understand how they're operating, giving us understanding that could lead to clinical use of these brain organoids. So this is why researchers are engaged in this work. It's actually, this is not a Frankensteinian attempt to create some kind of bizarre entity, some kind of cyborg. There's actually legitimate reasons why researchers are doing this work. Now, there are limitations right now. One is that the size of the organoids is limited, which means you only have about 100,000 neurons to work with, which is a very limited neural network. And the hope is that one day we should be able to grow larger and larger brain organoids, which means more neurons and more processing power, more capacity to store memory and store information. The limitation, again, is how do you get nutrients to the interior? People are exploring the use of microfluidic technology to create these artificial blood vessels that could feed nutrients to the interior of the brain organoid, allowing it to get much larger, with the hope that maybe we could create brain organoids with 1 million or 10 million neurons in them, dramatically increasing the computational capacity and the capacity to store memory. Another limitation is these brain organoids live for a period of time before they die, and so would have to be constantly replaced. They have to be bathed in nutrients, in a media to supply the nutrients and to remove the wastes. This is another technical limitation that has to be overcome. And then I also have concerns about reproducibility of growing the same kind of organoids over and over again that have precise, carefully controlled, high-quality capabilities. That's something else that has to be overcome. And so right now, everybody that's working on this is largely working on it as a proof of principle in terms of its technology application, in terms of biocomputing, but in terms of developing a fundamental understanding about brain structure, brain growth and development, how these might be utilized in a clinical setting, that work is much closer to some kind of practical realization. So the bottom line is there are very good reasons why researchers are doing this work. Now, what does this mean for the Christian faith? That's really the question I think many of us are interested in. Well, first of all, there's a very powerful argument for design in this work, because what this work is highlighting is, as remarkable as neural network technology is, and the production of neural networks by computer scientists and electrical engineers, as remarkable as that is, there are very clear limitations that researchers are running into now in terms of energy requirements. And yet it's remarkable that the human brain, with somewhere between 86 to 100 million neurons and an astronomical number of neural networks in interneuronal connections, has such incredible computational power, has such incredible capacity to store information in the form of memory, and yet utilizes, again, a very low level of energy. That just highlights the incredibly elegant design that we see in the human brain. And so from that standpoint, this work, again, I think can be used to make an argument that not only the human brain, but the brain of animals really does look like it is the handiwork of a creator. And in fact, I've developed an argument that I call an argument for design from biomimetics. And the idea here is that the human brain, and really the brains in general, have inspired a technology called neural network technology, right? And so for the brain to inspire a new technology means that it must be elegant, it must be sophisticated in its design. Why on earth would you turn to a design that's been cobbled together through unguided evolutionary processes to develop new technology? But if indeed the brain is the work of a mind, it makes perfect sense that you would use it to develop new technologies. So I would call this the argument from biomimetics or bioinspiration. And I see, again, this work on organoid intelligence is really highlighting the incredible designs of not only the human brain, but animal brains as well. But now, in terms of this larger question, should we play God? What should our Christian response to this technology be? And to me, the answer to that question really is found in the biblical concept of the image of God, that as human beings, we bear God's image. And theologians debate as to precisely what is the image of God. I'm not going to get into that debate here. But because we bear God's image as human beings, we have been given certain responsibilities by our Creator. We are to multiply and fill the earth. We are to subdue the earth and bring it under our control. And these two commands really relate to the idea of promoting human flourishing. We want humans who bear God's image to cover the earth. We want to bring the creation under our control in order to take advantage of the providence that God is embedded within the creation of itself. By understanding the creation, we're unleashing that providence in the form of scientific insight and technology that promotes human flourishing, human advancement. We are to rule over the planet because we bear God's image. God has made us his vice regents here on earth, given us the responsibility to not only rule over his creation, but to be stewards and caretakers of the creation. And so in light of this, we could see that work in this particular area helps to promote human flourishing. It's leading to technological advance that's going to drive economic growth, that's going to improve the quality of people's lives. It's going to give us fundamental understanding about how the human brain works, how the human brain develops, which can lead to biomedical applications, including direct applications where these brain organoids could actually be used to treat people suffering from neurodegenerative diseases or from brain injuries. So it can be used for good to promote human flourishing. It's going to help us to care for the environment because, again, AI technology is requiring huge amounts of energy in its current state. And this energy is potentially causing damage to the environment, the need for that high level of energy. And so if we can come up with technologies that use a minuscule amount of energy compared to current technology, that is a way in which we can be better stewards of the planet. So I see very good reasons why as Christians that we should be in favor or support of this work. Now, there, of course, is a counter to that, and that is that as human beings, we recognize that we also suffer from sin and that we can abuse technology just as well as take advantage of technology using it for good, that we can use technology for bad. And this is why it's very important for Christians who have been redeemed, who have the capacity as new creatures in Christ to overcome our sin nature, to be intimately involved in this kind of work, to have a good understanding of the work so that we can participate in the debate that takes place in our culture at large as to how this technology should be used, that we have a credible voice. If we understand the technology, our voice is going to offer an important perspective that's going to guide the appropriate use of the technology. We also need young people in the church who are interested in the life sciences, who are interested in engineering and computer science, and who are interested in biomedical research to get involved in this kind of research, being salt and light into a community that desperately needs Christian perspective to guide it in terms of developing this emerging biotechnology. And so while indeed we are sinners, we also are redeemed as Christians and have that capacity, that wherewithal, through the guidance of the Holy Spirit and the power of the gospel, to actually step into very complex areas and make wise, ethically sound decisions that really do indeed promote human flourishing while caring for the planet. There's something else that as Christians we also need to consider as well, and that is the potential that these organoid intelligence systems may actually develop self-awareness, right? May actually develop a type of consciousness, if you will. And right now, the technology is so primitive, I don't think that is a concern, but this is something that we need to keep in the back of our mind as we see this technology progress and develop, particularly as these brain organoids get more and more complex. But in effect, this is really no different than the debate that we would be interested in as Christians regarding artificial intelligence and whether or not those systems can actually develop self-awareness and a consciousness and even perhaps a type of free will. So that concern is really no different than what we would face anyway and what we are facing anyway when it comes to artificial intelligence. So it's a complex area, to be certain. It is bizarre science, but it's also at the same time fascinating science as well that highlights the elegant design of the human brain and really provokes a critical question: should we play God? And the image of God concept really helps to guide us as we think about this technology in a way that, again, is, I think, appropriate in terms of bringing glory to God. When it comes to the image, playing God, I always argue that as image bearers, we have no choice but to play God because we bear God's image. We are creators in the sense that our God, in whose image we are made, is a creator as well. And the concern isn't, are we playing God? The concern is, are we trying to take God's place? And it's only from a Christian perspective where we're certain that that's not going to happen. Okay, well, I'm very curious what your thoughts are about this technology, what your thoughts are about my response to this emerging biotechnology. Use the comments section if you would like to offer us your perspective, your thought. I'm dying to hear what you have to say. And so I'm going to go ahead and bring everything to a close by just asking you to make sure you visit our website, reasons.org. Consider making a donation to support Star Cells and God. Also, I would ask you to follow us on social media and make sure you go to our YouTube channel, Reasons to Believe, subscribe, use the notification button. Until next time, I want to leave you with this final thought. The more we know about science and the more we know about technology, the more we have reasons to believe. God bless you.